Well, we are here again with our second talk of the day. We are receiving Dr. Andreu Rico from the Aquatic Ecotoxicological Group of the Instituto Madrileño de Estudios Avanzados in Madrid. Andreu obtained his PhD on environmental science at the Wageningen. It's too difficult to say this word. Wageningen University in Netherlands. He's involved in several projects which aim to assess the environmental risks of agrochemicals and other emerging contaminants, such as pharmaceuticals and home care products, by conducting experiments both in laboratory and in the field. Even before your talk, I already have a question, but I can wait until the end. And so let's hear the talk. Thank you for accepting our invitation. Thanks a lot.
Thank you very much. I'm not sure if I should say good morning, good afternoon, but I just say good to everyone. And I'm going to share my screen so you can, from now onwards, see my talk. Okay, I hope you can see my screen now.
Yes, it's working.
Perfect. So thank you very much again for inviting me to giving this lecture. I entitled it Ecological Risk Assessment of Chemicals, Current Situation and Future Perspectives. And I want to give an overview on methods that are used to quantify the impacts of chemicals in the environment and how I see possibilities to improve the ecological realism of our assessment and our, let's say, overall robustness in the risk assessment. First of all, I just want to give you some information about who I am. I'm a Spanish person of 37 years old. I'm father of two girls, which take most of my time. And after this, I also do some research. I like traveling and observing nature, and this is probably the reason why I'm doing this kind of work, and you will understand later. Regarding education, Julian has already introduced me. I have done a bachelor in agricultural engineering and environmental science at the Polytechnic University of Valencia in Spain. And then I moved to the Netherlands, where I studied my master in aquatic ecology, and then I did my PhD on aquatic ecotoxicology. Then I moved to the IMBEE Water Institute in Spain as a postdoc researcher, and nowadays I'm the principal investigator of the ecotoxicology group. So most of my research is on ecotoxicology, spanning from environmental chemistry to risk assessment, and it is mainly dedicated to water because I work in a water institute. So I focus on aquatic ecosystems, and the focus of this talk will be on aquatic ecosystems. I don't like to talk much about metrics and CVs and things like that, but well, just to point out that I've published more than 50 papers and I have an Edge index of 21 in Google Scholar, and I currently supervise 4 PhD students. I lecture in 2 masters at the University of Rey Juan Carlos and the University of Alcalá in Madrid, and I've participated in more than 15 projects. I'm a member of the Society of Environmental Toxicology and Chemistry, and I'm an editor of the journal Environmental Toxicology and Chemistry. I work along 3 lines of research. I look at the environmental effect and effects of antibiotics, mainly in aquaculture production scenarios. So I try to understand how the antibiotics move from the fish into the aquatic environment and how these antibiotics may pose risks for non-target species, but also how these antibiotics may affect microbial communities and the development of resistance in these environments. I have also another research line on the fate and effects of microplastics in soil and freshwater ecosystems. So I try to understand how microplastics are retained by wastewater treatment plants, and how the distribution of sludge from these wastewater treatment plants into agriculture brings, again, these microplastics into the environment. And I try to understand how these microplastics move from the terrestrial to the aquatic ecosystems by runoff processes, for example, and what are the impacts of these microplastics on living organisms. And the third research line I work on is the risk assessment of chemicals, organic chemicals, and I try to understand how to prioritize compounds that are posing a risk for aquatic ecosystems, and I try to investigate the combination of the risk posed by chemicals and other stressors on populations and aquatic communities. So how these factors interact, how populations decline, and what is the possibilities for recovery after stressors have decreased and sort of like that. I work in the lab, but also in the field by doing monitoring experiments and mesocosm experiments and microcosm experiments. And this last research line will be the one I will put more emphasis to develop the talk I will give today. So I thought it will be good for people that is not really familiar with ecological risk assessment to start by learning some of the basic methods we use to prioritize compounds that are posing a risk in the environment. Then I will continue describing some novel approaches for the selection of vulnerable taxa to chemical pollution. And I will finally conclude my talk on integration of ecology and environmental science into risk assessment. So how can we integrate what we know about interactions between species or other factors on ecological risk assessment of chemicals? Just as a housekeeping message, I put this non-tweet tag in the bottom of this slide, which means when you see this, please don't take pictures or don't share this information because it's just work in progress. And of course, we don't want to that this is spread before this work gets into the journal. So please keep this in mind when you see this sign. So I would like to start my talk with a challenge. The challenge is that we live in a world that is dominated by humans, and human activities contribute to the emission of more than 100,000 chemicals into the environment. And at the same time, we luckily live in a planet that is inhabited by more than 8 million of species. Of course, many of them are undiscovered. So this is just an estimation. But when we put these two numbers into a kind of equation, it is very clear that it's very likely that some chemicals will pose some ecological impacts in ecosystems. And we need to identify which are the chemicals out of this plus 100,000 that are really posing a risk to identify management actions to abate them in the environment. But we also need to identify which are the components of ecosystems or species that are mostly affected by these chemicals. And this is the task that will take us during this talk to try to understand this. Ecological risk assessment is generally defined as the capacity to assess the possibility, if we talk qualitatively, or the probability, if we talk more quantitatively, that some adverse effects occur in ecosystems due to the exposure to one or several chemicals. And yes, so we need to understand what is the exposure in the environment of these chemicals, try to understand what are the mixtures that are there in environmental compartments, what we usually call the exposome. But we also need to understand how these chemicals can affect different biological endpoints. And it makes clear that this ecological risk assessment paradigm is based on two phases: exposure on the one hand, and effects on the other. And finally, we try to combine them to estimate a probability that effects occur in the environment. I would like to introduce a little bit some basic concepts. So for the exposure assessment, we talk about predicted environmental concentrations, which are those concentrations of chemicals that are emitted into the environment and for which we can predict peak exposure concentrations over time. And this is usually done based on environmental models and scenarios. These are models that have been built by people working on hydrology, environmental science, mathematics, and that are available nowadays to estimate peak environmental concentrations. On the other hand, we have the possibility to go into the environment, take a sample, bring it to our laboratory, and measure, based on advanced analytical techniques, what are the contaminants that are there and what are the concentrations of different contaminants. That's what we call measure environmental concentrations. And this is not new. You probably have heard about these techniques, and you have seen studies where they report environmental concentrations of a wide range of chemicals. On the effect assessment part, we try, as I said before, to understand what are the effects of chemicals to different biological endpoints. So the easiest way to do that is just to perform a toxicity experiment or a toxicity test in the laboratory. So we can put several organisms exposed to different concentrations or doses of a given chemical, and we can observe, for example, mortality, effects on reproduction, on growth, etc. Based on this kind of tests, we can define what we call a no observed effect concentration, which is the highest concentration that has no significant effects as compared to the control. Or we can build dose response curves by fitting all this data and bringing a curve in between, and we can derive the EC50 or EC10, so the effective concentration for the 50% of organisms or for the 10% of organisms. I'm sure you've heard about these concepts during your studies, and this is not complicated. This is just our way to assess the sensitivity of organisms to different chemicals. If we look at databases nowadays, and at the most regulatory part of this risk assessment, for most chemicals, there are tests or data that has been produced for assessing effects on algae, on Daphnia magna, as representative of invertebrates, or model organisms of fish. And if you go into databases, you will usually find at least 3 toxicity values for every chemical, which are commonly used for the registration of substances in many countries. But there are many other tests performed with other species that are not necessarily these standard model species that also help us to understand what is the variation in sensitivity across species and across trophic levels for different chemicals. Well, and now the challenge is to combine these environmental concentrations, predicted environmental concentrations or measured environmental concentrations, with effect concentrations, EC50 values, for example, to calculate some sort of risk or impact. In deterministic environmental risk assessment, we usually work with one endpoint, one biological endpoint, for example, an EC50 for Daphnia magna, and one environmental concentration, and we are capable to calculate toxic units or risk quotients that are also called in the literature. But the reality, as I said before, is quite complex. There are several substances occurring at the same time, so we can also implement these kind of equations to calculate the toxic units produced by a combination of chemicals in the environment by assuming concentration addition of several chemicals. And based on these basic calculations, we usually can classify whether the exposure is expected to make a moderate risk or a high risk, depending on the size of this toxic unit. Take into account that the fact of having this toxic unit above 1 is usually considered a high risk, but also 5, 10, 20, 100 is still a high risk, but this increase in toxic units does not tell us much about what is actually happening in the environment, what is actually happening to these species or to the ecosystem. So it is a kind of semi-quantitative method. In probabilistic risk assessment, what we do is to try to integrate as much toxicity data as possible for every compound. So in this way, we can calculate the percentage of a species that is expected to be affected by one given concentration of one substance. This is done on models called species sensitivity distributions, which integrate EC50 values, for example, for all species available for one single compound. So by entering this probabilistic distribution with one value for one environmental concentration, we can go to the y-axis and calculate the potentially affected fraction of a species. And of course, this distribution has its mean and standard deviation that characterizes the sensitivity of the taxonomic group and the variability in sensitivity in the species within the taxonomic group. When we have several substances, we can implement the same concept of multiple of the potentially affected fraction, and then it's called multi-substance potentially affected fraction. And this can be calculated with these formulas I give you here. I don't want you to understand them. I just want to let you know that it's possible to estimate the fraction of a species that will be affected by a combination of chemicals in the environment. And usually, a threshold is taken as 5% of a species. So when concentrations affect to more than 5% of a species, we consider this a high risk. And when it is between 1 and 5% of the species, we consider it is a moderate risk. So differently to the toxin unique approach, we now can estimate quantitatively what is the impact on the species that inhabit our ecosystem. Now I want to give you an example, and I thought it was very interesting to talk about one example of one study we did in Brazil last year. The paper is in preparation, so please don't share this information, don't take pictures and so on. And we studied the distribution of pharmaceuticals in the Amazon River. And besides pharmaceuticals, we also included other drugs and home care products. And we look at a total of 43 compounds in 40 sampling sites, spanning from the Amazon River in Manaus down to the mouth in Macapá and Belém. And we took samples in the Negro River as well, in the Tapajós River, in Tocantins. And we also took samples in the tributaries or igarapés in Manaus, Santarém, Macapá, and Belém. And our hypothesis is that urbanization in these areas is really contributing to an increase of pharmaceutical exposure in the environment. And we wanted to assess what was the risk of these pharmaceuticals for Amazonian freshwater biodiversity. We did our risk assessment based on the MSPAF approach, so the multiple substance potentially affected fraction approach, with acute and chronic toxicity data for all organism groups together. And as I said, our objective was to identify compounds that are posing a risk for biodiversity, but also try to identify pollution hotspots in this area. And these are the results of our investigation. On the top, you see a panel where you can see the different potentially affected fractions of a species for the different samples we took. You see that the exceedance of the 5% threshold occurs for acute exposure in Manaus and Macapá and Belém. And the main drivers of toxicity there were strong paraxanthine, which is the metabolite of caffeine, and furosemide, which is an antihypertensive. We did the same with the chronic toxicity data, and we managed to identify much higher risks. So about 50 to 80% of a species will be affected in the same areas, in Manaus, Santarém, Macapá, and Belém. And in this case, for a chronic risk assessment, caffeine, paracetamol, estrone, and ibuprofen were the main drivers of toxicity. So what we found in this study was that maximum accumulation or maximum exposure of pharmaceuticals occurred in the areas next to the main urban cities or to the urban regions of the Amazon. In the main river, there is so much dilution that despite we saw pharmaceutical concentrations at trace levels, we are not expected to find a risk for freshwater biodiversity. And yes, in the urban areas, freshwater biodiversity is severely affected. So these are hotspots of contamination. And just to conclude this first section, I just want to repeat that the environmental risk assessment methods allow the identification of chemicals and mixtures that are potentially affecting biodiversity and to identify hotspots, as I saw for the Amazon. What we found from our studies is that only a few compounds drive the risk. So despite we see complex mixtures in terms of exposure, when we perform our risk assessment, only 3 to 5 compounds are the actual drivers of toxicity and of biodiversity loss. And this is consistent in several studies done in other parts of the world. So this is good because this means that at least the complexity we have in the environment when we transfer it to biodiversity loss is much more reduced. And I saw methodologies for the calculation of risk assessment based on the toxic unit approach and the MSPAF approach, which can be complementarily be used, but the MSPAF approach offers a more realistic and more ecologically relevant estimation because estimates quantitatively the percentage of species that may be affected. The limitation of the MSPAF approach is that you need several toxicity data points to build the species sensitivity distributions, and several times for many compounds, we don't have such amount of toxicity data. So when toxicity data is not available, rather than eliminating a compound from the assessment, it's preferable to evaluate it under the toxic unit approach to at least have some idea on what is the potential impact for the ecosystem. And well, I've talked about methods to try to reduce complexity in terms of exposure, to narrow down or prioritize several compounds that are impacting ecosystems. But the other side of the coin is that we have several species, and we have also to identify what are the species that are most likely affected by chemicals. And the question we can pose is, can we identify a priori sensitive species, or can we learn about the sensitivity of a species even before testing it? So what we know is that difference in sensitivity between species to chemicals are a function of differences in toxicokinetics and toxicodynamics. And I explain a little bit. Toxicokinetics are the processes by which species uptake chemicals from the environment, biotransform or metabolize them, and eliminate them from their bodies. Toxicodynamics are the process by which these chemicals damage the organism of its species or the metabolic function, and the processes by which these species try to repair this damage or adapt to this damage. So these toxicokinetics and toxicodynamics are really the key to understand why, for example, a daphnid is more sensitive than an ephemeroptera, or the other way around, to different chemical substances. And how can we better understand this relationship between toxicokinetics and toxicodynamics and sensitivity? So how can we establish a pattern that allows us to quantify differences in sensitivity? So what we propose is to work with traits, biological traits, which are a phenotypic or ecological characteristic of an organism. They are usually measured at the individual level, and they can be physiological, morphological, or ecological. So for example, the fact of having gills may make that some organisms have a higher capacity to uptake certain substances than other organisms that don't have gills and breathe by tegument or by aerial respiration. The fact of having a more sclerotized body or not, or more lipidic body, can also contribute to higher bioaccumulation of chemicals. So all these little features that characterize our biodiversity can contribute to modify toxicokinetics, toxicodynamics, and at the end affect sensitivity to chemicals. So in 2012, Rubak et al. Published a very interesting paper where they studied the correlation between traits and toxicokinetic parameters measured from laboratory tests. So they measured traits and toxicokinetic parameters for 15 invertebrates and the insecticide chlorpyrifos. And they found that about 50% of the variation in the uptake parameters, about 85% of the variation of the uptake parameters of these species could be explained by 4 traits, and about 75% of the variability could be explained by other 4 traits. So we can already define species that have these traits and that are likely to be sensitive to one chemical or another. And based on this idea, we did a follow-up study where we studied the correlation between traits and sensitivity for macroinvertebrates for different insecticide classes based on EC50 values collected from available toxicity databases. And we built our regression models between sensitivity and traits. I show you here an example for organophosphates. So by knowing the size, the sclerotization, and the fact of being an arthropod or not, we can enter into this equation and calculate the relative sensitivity of a species to organophosphate insecticides. In a follow-up study, Van der Berg et al. Followed the same principle, but they also included taxonomic descriptors. And by including taxonomic descriptors, they could even improve the R-square of these fitted models. So now, with description of traits and taxonomy, we can estimate whether a species is expected to be sensitive or not to a specific chemical or chemical class. But well, everyone that works in ecology or in biology know that sensitivity is only a characteristic of resilience of ecosystems, but recovery is also important, especially in the long term. So what we did was to try to create an invertebrate vulnerability index. So if we can, based on traits, produce an estimation on whether one species is sensitive or not, we can also add a component to this equation that is based on several traits involved in the recovery, and in this way, estimate the vulnerability of a species to a specific group of chemicals. So for example, traits that are involved in recovery include volatilism, the capacity to recolonize via air or via water, or the capacity to swim and to move from one ecosystem into another, etc. So based on this equation, we can now distinguish vulnerable species from less vulnerable species to specific chemical classes by traits. So no testing organisms, just by knowing their physical, morphological characteristics and ecological characteristics, of course. And this altogether allows us to make a sensitivity and a vulnerability ranking, of course. So here we see for a wide range of invertebrate groups, how they rank in terms of sensitivity and vulnerability to pyrethroid insecticide, for example. So we can see that Odonata, for example, is the group that is more to the left, so is the most sensitive group. But on the other hand, is not the most vulnerable group. Megaloptera was found to be the most vulnerable taxa to this specific group of compounds. And this has severe implications for management, because often we want to protect sensitivity when compounds have a short life in the environment, but when they have a long life, or when we want to look at impacts in the long term, we may be more interested in vulnerability. Let's say Odonata, being the most sensitive, can be really affected by pyrethroids, but will recover faster. However, Megaloptera might be a bit less sensitive, but will take probably 1, 2, or 3 years to recover. And this is another interesting study because they use these principles, these regression models with traits, to classify the sensitivity of invertebrates in different regions of Europe. So they took monitoring data representing different regions of Europe, and they assessed the sensitivity to different chemical classes based on their traits. And then they found that, for example, in the south of Europe, we have a higher prevalence of sensitive invertebrates to narcotic compounds and to organophosphates. And of course, this has implications for management, because then this means that potentially the impact of some chemicals is larger in some regions as compared to others. And just to conclude this part, I want to repeat that a large part of sensitivity variation can be explained with biological traits, and we can include taxonomic descriptors to improve this correlation. The part of variation that is not explained is probably related to the toxicodynamics part, for which we have very limited information, so we don't know exactly how we can approach it, how we can model it, because we don't have clear evidence on traits that relate to toxicodynamics. Traits or trait-based approaches can help us to identify sensitive and vulnerable taxa, as I have shown. So they can help us to prioritize areas as well with higher predominance of sensitive or vulnerable taxa to certain chemical types. So now we've traveled from the complex area of many chemicals, narrowing down to few that really make a risk for the environment, making use of risk assessment methods. And we have implemented a little bit of trait-based approaches, ecology, into the identification of species that are potentially affected by these chemicals. And probably for everyone that is working in ecology will say, well, I find this is a bit simplistic because, in fact, species don't live alone in the ecosystems, but they interact among each other. Yes, this is true, and this is the next part of my talk. In ecosystems, we have several species interacting by competition, predation, etc., and therefore the impact of chemicals can be affected by these species interactions. And we have tried to go into this direction and investigate that a little bit. So this is a paper we published in 2017, where we look at the interactions between competition, predation, and an insecticide, chlorpyrifos. So in the first part, you see interspecific competition, where we have a control with so only Daphnia in vessels, and in the right-hand part, you have two treatments. One with Daphnia exposed to the insecticide chlorpyrifos at a very low concentration, and Daphnia exposed to chlorpyrifos together with Rotifera, which are supposed to be competitors for algae. And what we found here is that, interestingly, the sign of the interaction between the competition and the chemical was positive. So Daphnia were behaving or developing better when there were these competitors present. And then we found here that Daphnia was actually feeding on these rotifers. So it was a supplementary food source for them, so they could develop and reproduce much better, despite the effect of the chemical. So in this case, the effect was, the size was positive, which was relatively a surprise for us, and the strength of the effect was relatively small anyway. In the bottom part of this slide, you see predation. We have again the same setting. Daphnia was not exposed to chemical or to predators, and then Daphnia that was exposed to the chemical with a very mild effect of it, and predation with Caoborus larvae, which is an insect larvae that feed on Daphnia. And what we saw here is that Daphnia was much more affected when this predator present in the vessels. The sign of the interaction was negative and the strength was relatively large. And in this case, yes, Daphnia probably, the application of the chemical probably reduced the movement of Daphnia, so it was better hunted by the predator. So the predator had a, let's say, an improved capacity to predate on them. Now I want to show results of a paper published recently in Science of the Total Environment, where they investigated the influence of horizontal and vertical diversity on chemical effects. So will the effects of chemicals be larger or smaller when we have several species in the same taxonomic group or in the same trophic level? And will these effects of chemicals change when we have only one trophic level, or two, or three, or four? So we increase our vertical diversity. So keep in mind horizontal diversity, number of species within the same trophic level, vertical diversity, number of trophic levels in the ecosystem. And they did a relatively simple experiment where they put primary producers and they evaluated the biovolume of primary producers in test vessels, and they applied the herbicide Linron. We start with the left panel. In the left panel, we see two treatments. One in which there was only one species of primary producers, one species of algae, and the other treatment which had 5 species of algae. And they applied the compound Linuron, which is an herbicide. And they saw higher effects when there were when there was only one species than when there was 5 species. So the more horizontal diversity, the lower the effects as a whole. In the right panel, we see a treatment that there was only one species of algae and another treatment that had one species of zooplankton as a competitor, and the other treatment that had 4 species of zooplankton. So the number of competitors or predators in this case was increasing. And what they found here is that the effects of Linron were much larger when there was several predators at the same time in the system. So the more vertical diversity, the more trophic levels you include, okay, the more diversity in the upper trophic levels will increase the effects of the chemical. And to conclude this part, I just want to remember that the influence of species interactions depends on the mode of action of the chemical and the relative sensitivity of the species, of course, and this varies a lot, as we've seen before, so based on traits. Interactions are not always additive or synergistic, but often antagonism is observed. So often we see effects that are larger than what we expected based on the sum of the two stressors, but often they are much smaller. This is the example of the Daphnia with the rotifers I've shown before. And biodiversity and ecosystem complexity matters when describing chemical effects. So the more complex the ecosystem is, the lower the impact of chemicals in general. And this is a reason to protect biodiversity, of course. And well, probably people is seeing this graph in the right-hand side as missing one petal, starting to look like a flower. So let's make the flower complete. Yes, ecosystems are complex and impacts of chemicals are complex because we have several chemicals, several species. Species interact, but also species are affected by other abiotic stressors or anthropogenic stressors. Nowadays, we live in a world that the use of fertilizers is increasing, that the emissions of CO2 are increasing. We live in a world of constant change, climate change that is making an increase of temperatures, change in the whole hydrodynamics of ecosystems, problems of salinization in some coastal areas because of the lack of freshwater resources and changes in hydrological patterns and on rainfall patterns. So in this world of complex change, we have to take into account that these factors also influence ecological impacts. So let's see some examples on how these can be integrated or taken into account in our assessment. Here I show an example of a paper we published in 2016, where we investigated the relative contribution of chemicals to biodiversity variation in the Danube River. And so in the whole Danube River, I think more than 100 kilometers of distance, there were several samples taken and more than 300 parameters, descriptors of water quality, but also hydromorphological alterations and habitat characteristics were measured. And we did multivariate statistical analysis to understand what is the contribution of each parameter group to the variation in macroinvertebrate communities in the river. We found that habitat characteristics explain 16% of the variability, which is a big part. Hydromorphological alterations only explain 3%, and water quality parameters explain up to 31%. But if we look only at organic contaminants within these water quality parameters, only 8% of the variation was explained by organic contaminants. Most of the water quality parameters variation was explained by nutrient concentrations, pH, oxygen, and these sort of parameters. And what we found in this study was also a big, high correlation between the co-occurrence and the effects of these abiotic parameters. So this implies that chemical impacts are also coexisting with several other alterations of the ecosystem. And here I would like to show you an example of a paper that we published last year on how temperature, simply temperature, can alter the metabolism of organisms and the responses to chemicals. So in the top part, we have, we did this experiment with microcosms in the laboratory with several zooplankton species, and we tested two treatments, temperature at 20 degrees and temperature at 20 degrees. And within each treatment, we had a control, so with no chemical application, a treatment with application of lufenuron at 1 microgram per liter, and a treatment with one application of lufenuron at 8 microgram per liter. Lufenuron is an insecticide. At 20 degrees, for example, for Copepoda naupli, for the small stages of Copepoda, we found that there was a clear effect of the pesticide, and after several weeks, they started to recover. At 28 degrees, we find a similar trend, but we see that the effect of the pesticide occurs much earlier. So on day 4, after the first application, we already see an effect, whereas at 20 degrees, we see more clearly the effect at several days, almost more than 1 week after the application. And in the 28 degrees microcosms, we also find a faster recovery. So the whole metabolism of this population and the higher capacity to reproduce at higher temperatures makes that the uptake of the chemical is faster, the effect is faster, but also the capacity to recover is larger. And this holds true for the direct effects, but also for the indirect effects. If we look at the bottom panels, we see a rotifera and lecane species in this case, and rotifera really increase when there was a higher concentration of the pesticide, and the increase was faster at 28 degrees and larger at 20 degrees than at 20 degrees. So temperature alters the impacts of chemicals and also the indirect impacts of chemicals on the community. This is another experiment, very recent, it's just in paper is in preparation, by Jessica Andrade. And here we look at the interaction between temperature at 20, 22 degrees and 30 degrees, salinity, natural water conditions, and another factor of 2.5 grams per liter of sodium chloride, mimicking salinity intrusion in coastal wetlands in Spain. And we applied the insecticide chlorpyrifos at 1 microgram per liter versus the control. And we assess how these factors interact on the response of zooplankton communities over time. And we found in these microcosms that temperature, salinity, and chlorpyrifos really was making an impact on the community. Temperature and salinity were significantly interacting, and they were contributing to decrease diversity and abundance. So in a global warming scenario where there's also salinity, the impacts on the biodiversity will be much larger than in the current days. Temperature and chlorpyrifos also indirectly affected the response, but this was expected based on the previous experiment. Higher temperature, higher effect, and higher recovery. And salinity and chlorpyrifos didn't interact significantly. So we here found additive or rather antagonistic effects in the last sampling day. And we explained this based on the lower chemical bioavailability when there was salinity present. So what we see here is that these factors interact, modify the dynamics of populations and communities, and we should therefore take them into account in risk assessment. And I want to show a graph from a publication of Michelle Jackson in 2016 in Global Change Biology, where they did a meta-analysis of multiple stressor effects. And I focus here on the multiple stressor combinations that contain contamination. And they look at contamination versus habitat alteration, contamination versus invasions, contamination with nutrients, and contamination with warming. And they look at whether the responses of different biological endpoints were additive, antagonistic, synergistic, or reversal. Reversal means that the impact of one chemical is changing significantly the influence of the second. So it's a specific, or a little bit um, specific category that is not usually taken into account in many studies. And an interesting discovery from this meta-analysis was that most of the effects are found to be antagonistic. And what they discuss in this paper is that antagonism is usually um, a consequence of co-adaptation or co-tolerance. So when one stressor makes an effect that correlates with the effects of the other stressor individually, the application of one stressor can already select for the most tolerant species to the other stressor. And this way, this co-tolerance contributes to make the effect of the combination of stressors a bit lower than it was expected based on an additive model. But of course, it's very interesting also to see that in many studies, they find synergistic interactions. And this is important to take them into account because it means that in combinations of stressors, the impacts of biodiversity can be much larger than it was expected a priori based on the individual effects of chemicals. And well, I want to make a whole summary of my talk now. I just want to repeat again that the impact of chemicals on biodiversity is influenced by sensitivity and vulnerability of the exposed organisms, but also by species interactions and abiotic stresses. We should not neglect them. Current environmental risk assessment methods are effective for the prioritization of contaminants or contaminant mixtures that we've seen, and also can identify sensitive biological endpoints and areas where there is a high impact of contamination, but in the way they are created, they have a very high lack of ecological realism. As I said before, biological traits can aid in the identification of sensitive and vulnerable taxa that may be affected by chemicals, and we have also to take into account that the different mode of action can be correlated to different traits. Species interactions and biological complexity may positively or negatively influence the impact of chemicals, so it's important to take them into account. And further research is needed to understand the influence of multiple stressors, either biotic or abiotic, on chemical impacts. We are just at the beginning to understand the mechanisms and how these combinations of stressors actually work. But the next step is really to find a way to integrate them in the whole environmental risk assessment paradigm. And this requires more science and, for example, the development of models and thinking and specific experiments that integrate all this knowledge and all these hypotheses into a more holistic view. So this was my last slide. I really thank you for your attention. I hope that you like this talk, that you find something interesting or some ideas to discuss or to continue your work. And I will be really glad to answer your questions. Obrigado. Thank you very much.
Thanks a lot, Dr. Andrew. Well, there are some questions here already. I will wait to you for closing the presentation so we can see you.
Yes, I hope you can see me now.
Yes. So the first question is made by Jessica Andrade. She asks that, in your opinion, what is the main difficulty to predict using environmental risk assessment when we compare the tropical and temperate conditions?
Well, this is a very nice question because I've worked in this area a little bit. So there are several aspects to take into account. One of them is that we can hypothesize that sensitivity can be different, sensitivity to chemicals, because in the tropical region we have higher temperatures, so metabolism goes faster. So as we've seen before, when there is higher metabolism, there is higher uptake of chemicals, potentially higher elimination. So the effect becomes larger in the very beginning, but then the recovery is also faster in a way. So there are several studies that look first at sensitivity based on laboratory toxicity data and didn't find clear differences. And I think this is partly related to the fact that there's a lot of variability in the sensitivity of species in the tropical region, but also in the temperate region. So there are species that are more tolerant in the tropics than in the temperate region and the other way around. So as a whole, we cannot say that one region is more sensitive than the other. Okay. There are also microcosm and mesocosm experiments that look at whether the sensitivity thresholds can be extrapolated from one region to the other. And they didn't find systematic differences. You should also take into account that in micro and mesocosm experiments, the variability is large between the response of populations and communities. So as a whole, we cannot nowadays say that one region is necessarily more sensitive than the other. There are differences in metabolism, differences in some species and in diversity perhaps, but not systematic differences in sensitivity to chemicals. Which I think is important to take into account is that in tropical regions, usually the emission of chemicals, for example, I'm thinking now of pharmaceuticals, there's many areas where there's no wastewater treatment plants. So the emission of some chemicals can be much larger. And the fate or the degradation of chemicals can be very different because they have different light regimes, different temperatures, microbial activities. So I expect to see more differences in the terms of the emission patterns and the whole fate and exposure than on the sensitivity as we know today.
Carlos Moura asked the size of that silent spring. Since that silent spring, we are aware of this kind of impact, and unfortunately, we're still struggling to deal with it. The industry response was basically create new molecules based on if it's new, then it's better. Can we already track these new ones?
Can we, sorry, what was the last part?
Can we already track these new ones, these new molecules?
See, if we can track them, if we can, the question is if we can track them. So, yes, we should track them, that's for sure. I think there is a lot of work to do in terms of regulation and in terms of setting up rules for these new molecules. So we cannot simply create more new molecules and just use them for whatever. We have to follow a whole procedure of risk assessment before a molecule can be used and before it can be emitted into the environment. I know the case of Europe because I'm from Europe. There is more and more regulation nowadays, and by the way, more jobs and more people dedicated to this to really test new molecules. Starting by simple tests on algae, on Daphnia, whatever, very simple, but at least we have to provide a basic data set for every new molecule that we produce. Differently, and this is just my impression based on some of the work I've done in Brazil and some of the conversations with my colleagues from Brazil sometimes, the regulatory system has many possibilities to improve still in several developing areas or in Brazil, etc. So what we can propose is that we need to invest more. We need to provide more resources to test every single molecule that the market wants to bring to us and to the environment. It doesn't need to be a whole very complex risk assessment, but at least some testing to classify the molecule, to put them into a group, to make a basic risk assessment based on models, whatever. But this needs to be done. Otherwise, we end up in the risk of emitting substances that we don't know anything about them and that can damage different ecological endpoints that we are not aware of, for example. And I'm just reminding the case now of diclofenac. Probably you heard about this pharmaceutical that follow probably a very simplistic risk assessment when it was first used and emitted into the environment. And then after several years, they found that it was affecting vultures, and it was just an unexpected um, um, unexpected pathway or unexpected outcome pathway to effects. And the more we do to classify molecules and to see the potential impact on different ecological endpoints, the safer we are. So we definitely should work in this direction everywhere, everywhere, but especially in tropical countries, I would say, where there's often the regulation is not that developed or not that implemented.
Yes, your talk is a great alert that not just we must improve our behavior, but also to ask for public policies, and to the government must take providence to control, but to do a better control of these actions. The third question is made by Emilia Brovini. She thank you, and she asked, the risk assessment with multiple substances is calculated by the additive effects. Are there other methods with synergistic or antagonist effects, or is this still a challenge?
Yes, this is a very good question. So thank you very much, Emilia. It is indeed important to test different models of mixtures, like test for synergism, for antagonism. The reviews on this knowledge show that the cases of synergism are not many. So differently to what I've shown before for multiple stressors, where synergism was really occurring a lot, for chemical mixtures it doesn't occur that much. So we have to identify these cases, and for this reason, we have to do research into effects of mixtures for sure. But when it comes to the more holistic approach to assess the risk of a substance of a sample that contains 250 chemicals, often the concentration addition is taken as a proxy, as a way to simplify our capacity to predict an impact. And so my message is that this should be tested. Synergism has to be identified as much as possible. But when we have samples, and I've taken these samples in Spain and in many other parts of the world, that you have, or in Brazil, for example, in the Amazon, in the Igarapés of Manaus, Belém, you have 200 compounds there. So, and compounds for which you don't have almost information on toxicity. So you have to apply relatively simplistic methods to provide a sort of first estimate of risk. And once you have this, you can look, as I did, well, identified 3-5 compounds that are really driving the risk. Then you probably can do separate experiments with this small combination of compounds to test for potential synergism, antagonism, etc.
Well, now I will make you my question.
Yes,
last week here in Brazil, the environment minister, Alberto, our environment siniestro, allowed the use of a fire retardant over the Chapada dos Veadeiros. That it's a very important and wonderful area of natural protection. This product contains ammonium sulfate, and according to the technical report of IBAMA, which is a federal institution related to the environmental protection, the use of this product is not recommended. What can you tell us about the hazard and toxicity of these compounds on the on the ecosystem that is a hard ecosystem with a lot of hard species?
Well, that's interesting. I'm not an expert in this kind of compounds, so in flame retardants, but first thing I will do is to ask whether they have done any kind of risk assessment. Doesn't need to be complex, but have they gone to the field to measure this compound? Have they made any kind of test to see what is the impact of this compound on several organisms? So my recommendation is first test it, first try to do this kind of assessment, and later see if there is any options for replacement. Whether there are other compounds that offer a better alternative than this one. For doesn't need to be only on toxicity; it can be also on fate. For example, some compounds that degrade faster may pose a lower long-term risk to the environment. So I would say be careful in these cases to say this is not a problem if you have not conducted a risk assessment properly to support this. And in case you find some potential risks, try to find alternatives. Yeah. I know your president is not a fan of risk assessment.
Yes.
and this kind of research. But
well, I think that when there is a technical report of a federal agency that is destined to protect the environment that did not recommend the use, I cannot understand how something like this could happen. But that's why I always think that we must ask and ask and ask for public policies that will take care of us, the planet.
Yes, and I think we have to put, and this is good times because of the pandemic, because of coronavirus, is we have to put on the table again the value of science.
Yes.
So these reports it's not like a newspaper where you believe or not what the reporter is saying. Science is something else, something more serious, something that we try to do objectively all the time. And we have to try to, I'm not sure if the word is convincing people or just explaining people that science has a very high added value to respond to problems. And we are seeing to this with the pandemic, with the coronavirus nowadays.
Yes.
That really science is needed to solve problems of this size.
Yes, let's have hope. Andreu, we thank you a lot. It was amazing to hear and learn about the hazard and the risks we are exposed. Thanks a lot again, and thanks all for the audience.
Thank you very much for listening, and I really enjoy the questions. So I hope it was at some point helpful for you. Okay, thank you very much for organizing this event and to all of you. Thanks, Juliana.
Thank you. Bye, Professor.
Bye. Thank you. Obrigado.
So thanks everyone. Here's José. I just want to remind you that this afternoon at 2 p.m. Brazilian time, we will have our last symposium. And after the symposium, we will close the meeting. So we expect you all at 2 p.m. Thank you very much so far and have a good break.